Great to be here. Thank you for giving me this chance to talk about a bit about my research. Uh, it's the first time where I started to uh, I decided to start not with the title of my talk, but with the title of the session. And the reason is because I think that we are coming from many different fields and we have different backgrounds. So I would like to just mention for those that might enjoy the parallel session that I'm not going to talk about general theory of relativity or evolution, genetic, existential sense, and this kind of philosophical considerations in terms of origin of life can be really nicely found in the chapter our two questions in the PhD thesis of my friend Dario Lago. I'm not going to talk also about this James Webb Space Telescope new discoveries, which is very, very nice. This is a picture from 2023 I took from the web page. I will talk mostly about the chemistry origin of life. And I was inspired by this book published in 1993, where this magician, which is probably theoretician, is collaborating with different scientists with different background, experimentalists most probable, to really answer to this very complex question. So I will be talking about the peptide bond formation, and this peptide bond formation I will show you that can be induced by alpha particles in the clusters of beta-alanine. So as you know, the molecules are presented everywhere. So we are living in a molecular universe, and these neutral and ionic species there have been identified outside the Earth. Quite recently, in 2018, there was discovery or observation by this Cassini spacecraft on Saturn, Moon, and Enceladus, showing the presence of the complex organic molecules with the masses higher than 200 atomic mass units, and it's given series and so on. And we are interested in the origins, in the mechanisms of the formation of these complex molecules, which are believed to be needed for life. And I'm also interested in the effects of the ionizing radiation, so the damage to these molecules, and all this at the molecular level. So as you might see, the molecules of interest I'm gonna talk today is beta-alanine, and this amino acid was found in these carbonaceous chondrites, the meteorites that fall down in France and in Tanzania. These are the very, very primitive one. They are as old as our solar system itself, and they were already presented as the most abundant biomolecules, amino acids. So to try to understand this, the formation of these complex molecules, there are two approaches basically. One is called bottom-up, and this is very beautiful work published last year by Serge Krasnokutsky, where he showed that the atomic carbon can condensate on the ices of carbon monoxide and NH3. And he showed this in terms of the mass spectrometry experiments and quantum chemical calculations. The alternative approach of these growth processes can be considered as the molecular clusters. I call it a bridge. Bridge means that something that is bridging the world of the single individual molecules and the bulk or solid state physics. And here I presented a couple of papers that are interested to talk about. One, the one that I believe that everything started from coincidences. So from the measurements of the fullerenes in coincidences after interacting with these highly charged ions, like for example, alpha particles. And then some people later on showed the growth processes that can be induced by highly charged ions for biomolecules, fullerenes, pHs, which are polyaromatic hydrocarbons, polycyclic aromatic hydrocarbons, sorry. This pHs, mixture of pHs and fullerenes as well. So before I will start, I would like to tell you a bit about the computational methodology. So this was developed during my PhD. I was working with these three guys in Madrid. And in 2013, we published this methodology, this paper, basically based on the assumption. So if you would like to describe what happened to the molecular target when it collides with highly charged ions, represented here by this PQ+, we can do it in a statistical way. So we are basically assuming a Frank-Condon type transition. We are extracting outer electrons, and we give some charge and some excitation energies to the nuclear degrees of freedom of the molecules, target molecule, and we are propagating equations of motions from the molecular dynamic simulations, and we are getting the charge fragments. And with this approach, we are able to understand very different effects for different target molecules. From the theoretical point of view, what we are doing most of the time is running MD simulations. What we would like to, and this particular one, which is called atom-centered density matrix propagation. So we'd like to solve this Schrödinger equation, time-dependent Schrödinger equation. We are using some approximations without going too much into the details. I just like to say that we are basically using the Born-Oppenheimer approximation. So we are splitting this total wave function into nuclear and electronic wave function, and you can already see here that the electronic part depends parametrically on the position of the nuclei at a given time, and then we assume that these nuclei move classically. So this is so-called semi-classical method in the adiabatic potential energy surface of the given electronic state, and we are assuming most of the time that we are moving in a ground state. So this potential can be varied. We can use different electronic structure method to solve this. If you would like to learn more, go to these two papers, Schlegel and Carparinello. From the experimental point of view, what we are doing, we are collaborating with those people, which are shown here from Daniel CIMAP. They have this colimaçon end station. They do the cross-beam collision experiment. So you have a molecular beam in blue and ion beam in red. And you collide your molecular target. You apply some electric field. You are able to measure cationic products through this time-of-flight tube. It's a time-of-flight mass spectrometry. They are an experts in the coincidence measurement. So you can measure two charge fragments coming from the same event. And they also have a nice gas aggregation cluster source. So the full methodology looks like that. So we have the molecular dynamics. We run it at a given time in a statistical way, hundreds, thousands of simulations, usually at the FT level. Then we are taking this, we are building the potential energy surface, and then we compare what we have in common, which are the charge fragments. This is the coincidence mass spectrum. So the time or mass over charge ratio as one fragment as a function of mass over charge ratio of another fragment. And then from the theory, we get very valuable information like time evolution and energetics, and also the molecular structure. We can have a spectra like IR or Raman, whatever. So let's go to the results. So here there is the spectrum where our clusters collide, clusters of β-alanine collide with 30 keVs helium 2+. What we see here is pretty much the protonation of the cluster. So we see that the cluster survives, but they are protonated. And in the MD simulation, we see that this protonation happens often on the nitrogen atoms. So these are the three different trajectories. There is a time scale. So frames from the molecular dynamic simulation until 200 femtoseconds. So it's relatively quick proton transfer. And then what you can also see here, interestingly, not only that the proton migrates to the nitrogen part, but also that the cluster itself can fragment, which was kind of surprising that the covalent bond breakage can happen within this cluster, keeping the rest, this protonated cluster, stable. So this protonation stabilizes the cluster. What we also see, what is more interesting, are these intermediate peaks. So the peaks between these protonated clusters. And these are the fingerprints of the covalent bond formation. And there is also successive loss of 18 atomic mass unit, and 10 is water, and water you might expect that is or might be related to the peptide bond formation. So can we really see it? To answer to this question, first of all, we try to understand from the experimental point of view if we have the same mechanism. So here I'm comparing the spectrum, the mass spectrum between the monomer and dimer and trimer. And you can see that there are similar patterns, especially this marked in the orange. So to answer to this question, what can happen, we run another quantum chemical calculation. So here on the upper part, you see the potential energy surface. So we have this protonated dimer that we start from molecular dynamic simulations, and so with a very, very low barrier, we can form the peptide bond formation. The energies are in electronvolts, so it's very low in energy in respect to the first vertical ionization potential. And the same mechanism, starting with this saddle point, we see from the molecular dynamics simulations, so also peptide bond formation, which has mass 161. And when we start building the potential energy surface for this singly charged species, we can easily explain peaks that are also very strongly observed in the experiments. We can get more information. I'm not going to talk about in details, but as you might see, we can, for example, see where are the exit channels, so-called exit channels, when the further subsequent fragmentation with the same mass overcharge or with the same masses happens. So, for example, like for the hydroxyl and for NH3, so you can see that the breakage on the NH3 part is more favorable from the energetic point of view than breakage of the hydroxyl group, for example. But we can also see something interesting when we study the doubly charged protonated dimer, starting from here. Then we can see easily formation, again, of the covalent bonds with a subsequent fragmentation. And then it will be very cool to see it in the experiments, in the coincidence experiments. For example, 45, so this fragment, and the rest of the fragment is 134, which was not possible to be explained from the singly charged protonated dimer. For this, we need charge, so we need D cations, and then you can see that if you plot it, this is the same type of a spectrum. So this is a coincidence spectrum, but plotted in a different way. Basically, you can see, for example, what are the fragments that 45 is measured in coincident with, and 134 is the most abundant peak in the experiment. So this is like the final experimental evidence of covalent bond formation within the clusters of doubly charged clusters. So I'm a little bit too quick, sorry for that. Basically, I would like to conclude. So what you could see here is that the helium-2+, so alpha particles, can induce the origins of life reactivity inside of these clusters of β-alanine. We use also the strong stabilization by protonation, evaporation of neutrals. We also see that the barrier for the peptide bond formation is very low. I didn't show this, but we see exactly the same for trimer, like in this scheme. So we see evaporation of neutral residues, formation of a protonated trimer, and then the formation of the peptide bond within the trimer. We also see that this doubly charged cluster can form different covalent bonds, and this can be, without any doubts, confirmed by the ion pair coincidences in the experiments. I would like to acknowledge, so two most acknowledged people are Patrick Rousseau and Sergio Diaz-Tendero. Part of the work I've done regarding this work was done already during my postdoc in Münster, Germany, when I was working in the group of Professor Olga Garcia Mancheño. And I would like to thank to the leader of the group I joined after Germany at Polish Institute of Physical Chemistry in Warsaw, Adam Kubas. And the last slide is the advertisement. So if you liked the approaches I showed you today, please drop me an email. The deadline for the application is 24th of, it's 4th of January actually. The application will be open between 24th of January and December and the 4th of January. I have a position for a PhD student to work with me and also with a postdoc that is going to build this nice experimental setup. This will be also work supported by our colleagues from different leading European centers from both experimental and also theoretical point of view. So thank you for your attention.